This Bio 181 video is on cell signaling, chapter 9. We're talking about receptor proteins and just their location. So they can either be on the surface of the cell, so on the membrane. So you can have your receptor protein out here. So this is a transmembrane protein. It's going to be in the membrane in the phospholipid bilayer. So it can be out here or it could be inside the cell. Okay. So again, outside the cell, things that are not allowed in the cell are going to be molecules that tend to be polar. So they have a charge imbalance, or things that are charged. So have an entire full +1 or -1, like an ion charged or even more. Polar is just an imbalance of charge. So, or a molecule that's large. Right? So molecules that are going to fit this category are often going to be proteins, just small little proteins, but proteins are often, they have different R groups or side chains that may have polarity and or charge. So molecules outside, this would be something like insulin, which is a hormone that is a protein, right? This is a protein. So it's going to bind outside. And then things that can diffuse through are things that are nonpolar. So the thing that we went over in this class a lot are steroids. So steroids are a class of hormones that are lipid-based. So they're made out of lipids. So this would be testosterone, estrogen, cortisol, which is a stress-released hormone. And so those, they're these little chains, they're basically 4-ring structure lipids. Lipids don't all look the same, but they're all nonpolar. So these are going to go straight through. So maybe in this case, testosterone is going to go in here. And then it's going to bind to the receptor. So if the receptor in here would have, a complementary shape on it, let me see if I was to draw one kind of like this, it might have a shape. Well, let me copy this. So copy, okay, paste, paste. All right. Oh, I wanted the whole thing to go in. So, but this whole molecule would come in, and I'm just going to erase this other receptor. The receptor would have a complementary shape on it in here. Of course, this is not to scale, right? But this would be my receptor molecule in here. So it'd be located in the cell because this would diffuse in. Of course, it would still have those other rings on it. I don't want to take too much time here, but you'd have this 4-ring structure. It would bind inside the cell. All right. So receptors include channel-linked receptors, enzymatic receptors, and G protein coupled receptors. So let's go over each of these. So if I have a cell membrane, I can actually have say an ion channel embedded in the membrane, and it's going to open in response to receiving a signal. So may have a portion of it over here. Okay. Let me just erase this a little bit, where there is kind of like an active site on an enzyme. There's a portion that is going to receive a signal, and sometimes it might even take 2 signals or 2 ligands to trip it to go. But if we have a signaling molecule that binds there, so say here's my ligand, maybe I need 2 of these to open the channel. If these 2 bind, then all of a sudden this channel will open. Gosh, okay. I don't know if I can erase just a tiny amount here. Doesn't look like I can. So let me not try to do that. But the channel will open and then it will let in any ions that that channel is made for. So, all right. So let's say sodium channel, sodium. And this channel might open in response to receiving these signals. All of a sudden, sodium, if there's a whole bunch outside, it will pour into the cell in response to this ligand binding. And this is how we send nervous signals, really quick electrochemical signals through the body. You have a neurotransmitter, and it's going to bind to an ion channel. That channel is going to open. You're going to get flooded in with ions, and that's going to cause a cascading reaction. So that's a channel-linked receptor. So that's the first one. Then the second option here is an enzymatic receptor. So that means I'm on the surface of my cell. And now the protein receptor is an enzyme. So I'm just going to draw a blue enzyme. So now we're talking about the enzymatic receptor. So over here, I've got this molecule is bigger than it would be realistic, but say this is my enzyme. Okay. And so I've got a signaling molecule. The signaling molecule binds to the enzyme, right? This is my ligand. Again, a ligand is going to bind here. This is my ligand. It's just a signaling molecule. So is this. And it's going to catalyze a reaction. And so often that reaction is actually going to be catalyzed on the inside of the cell. So this part over here might change its shape in response. So once the ligand binds here, then this internal part of the enzyme might change its shape, and it might catalyze the reaction in here of, say, adding a phosphate group to something. So maybe it's going to catalyze the addition of a phosphate group to a molecule. That's going to energize that molecule right in here. And so now it has this phosphate group attached to it, and it can go and energize a whole bunch of other reactions. So an enzymatic receptor, the receptor itself is an enzyme. And then the last category is a G protein coupled receptor. This is so common that we are going to talk about it. So as just a specific example, so you've got a protein, let me just different color here. So you've got a receptor that's in the membrane. Okay, here's my receptor in the membrane. I'll make it very generalized. Okay, and again, it's going to receive a signaling molecule. So say there's a part here. It's going to receive a signaling molecule, orange ligand. Okay, and if this happens to be a G protein coupled receptor, this one, right, then what's going to happen is it's going to trigger or activate a G protein in here, a G protein, and that G protein often acts like an on switch, and it's going to move into the cell and usually turn something on. So it generally acts like a switch to turn on an activity. And again, G protein is just such a common receptor that we're going to give it its own category. All right, this is my last one, right? So I've got one more. So steroid hormones, last one for this video. Steroid hormones are hormones that are going to diffuse straight across the membrane, right? And so talked about this. They are nonpolar, so they're going to go in. I'm just going to make it simple this time. So I'm going to draw, here's my steroid. So say this is estrogen, and it goes straight through with simple diffusion. A steroid is a lipid, right? So it's nonpolar. So that means it goes through with simple diffusion. Okay. It does not need a protein channel. It's just going to go straight through the plasma membrane. And what's interesting about steroids is they often will bind to a receptor that's in the cytoplasm. So some sort of receptor in here is going to bind to it. Okay. And then this receptor is often going to move into the nucleus. So again, the nucleus is where we store our DNA. So you've got the DNA protected, safe in the nucleus. It has these pores that allow things in and out of it. So this is going to go inside, right? And this is, if it's going into the nucleus, this is called a nuclear receptor. It's going to take it inside. So the whole thing's going to go inside. And then it's going to trigger a reaction which looks for the DNA, which is our genetic code. And then the DNA is actually going to be used to express a gene. So to make proteins, which we'll talk about later, but you're going to take a gene and make something with it. So say it's estrogen, it might go in and say, all right, we're going to start making milk, right? If we're doing mammary glands and we have a like estrogen, we are going to, that's actually not a good one. That would be a different enzyme, but we are going to stimulate the production of secondary sex characteristics for females. So maybe it would just be breast enlargement. So then estrogen is going to come in, it's going to activate the genes that are associated with that, and then it'll trigger that response. Okay, that's